So hello everyone. So I will be starting the class for today. So I think the slides are visible, right? So I hope the slides are visible and my audio is also clear. So good evening everyone. So I am going to take the week 7 section on the course Cell Culture Technologies, which are conducted by NPTEL and the TAship that has been organized by IIT Madras. So I will just start with the first question itself. So question number 1. So white matter is mostly composed of which option? Option number 1, cell bodies, dendrites, shorter axons, myelinated axons, or cell bodies and axons in equal amounts, or none of the above. Which may be answered. Okay, so the answer to this question is myelinated axons. So I will just give a brief explanation of what is a white matter and why it is mostly composed of myelinated axons. So there's a figure here and also some description here. So you can see that this is a part of a brain, right? So it is a cut portion of the brain. You can see that it composed of different colors, right? One is a light brown color and other one is much darker brown. I am not saying it is much darker, but it's a light dark brown color, right? So basically you can see that these are two different types. One is a white matter, which is a lightly brown color, and most probably it is near to white color, and next is the gray matter. So basically if you look into white matter in central nervous system is primarily composed of myelinated axons, because you know what is a myelinated axons, right? Because there is two types of axons there. One is myelinated and non-myelinated axons there. So if in case of myelinated axons, there is a myelin sheath that covers across the space that is present within the axon regions there. If a myelin sheath is not present within the length of the axon, then it is called as a myelinated axon there. So if you look into the myelinated axon, what differs in case of peripheral nervous system and central nervous system is that in case of the myelin sheath formation, actually in case of peripheral nervous system happens in the presence of Schwann cells, while in case of central nervous system, that is in brain and all, as you are seeing here, it happens in the presence of oligodendrocytes. So basically what happens is that these Schwann cells actually in the presence of certain proteins called as myelin basic protein actually loses some of the boundaries that are present within cells. These are basically cells there, Schwann cells, and then they join together. Each and every cell, after losing their boundary in the presence of this MBP protein, loses its boundary, and they join together to form a sheet-like structure. So when that sheet-like structure is present, if a axon is present here, then they cover the entire axon to form that myelin sheath there. So repeatedly at different regions of the axon, then myelin sheath is formed and it covers each and every axon to form what is called as a myelinated axon. If these are not present there, then it is called as an unmyelinated axon. In case of central nervous system, the myelin sheath is formed in the presence of oligodendrocyte, which I will explain later in the coming slides. So this white matter is basically made up of myelinated axon, and because of the presence of this myelin, it is usually white in color. And if you look into a content or the lipid content that is present within each myelin, it is high in lipid content there. So because of this lipid content, it is usually white in color, and this through a process known as a saltatory conduction. In case of unmyelinated axon, then the conduction actually happens linearly. In case of myelinated axon, as the myelin is present in the different at different consequential space of the axon, it has to jump from one space to other to conduct the particular neural transmission there, and this particular process is known as saltatory conduction. So basically the myelin actually insulates the axons and facilitates rapid signal transduction, because the signals are jumping from one space to another, right? So in case of brain and spinal cord, it is essential in this case. So it plays a crucial role in communication with the central nervous system by connecting various areas of gray matter. You can see that gray matter is here, right? So in order to connect to other regions of the gray matter, then this white matter actually plays an important role there. So in the brain, white matter is located beneath the gray matter cortex. The gray matter is actually present in the boundary. Then comes the white matter. And in case of spinal cord, it is totally the opposite. The gray matter is actually present in the center, while the white matter envelops it. So I hope that this answer is clear. So anyone has any doubt? So I think the answer is clear. So I will just move on to the next question. So coming to question number 2. Gray matter is mostly made up of cell bodies, dendrite or shorter axons. Second option, myelinated axons, cell bodies and axons in equal amount, or none of the above. Which might be the answer? Okay, so the answer to this question is first option, that is cell bodies, dendrites and shorter axons. Gray matter is mostly made up of cell bodies, dendrite and shorter axons. So gray matter is primarily composed of neuronal cell bodies, dendrites and shorter axons there, right? So basically you have seen that in the previous slide I have shown you a picture. So this is the white matter, this is the gray matter that is present at the boundary of the brain. In case of spinal cord, it's totally opposite. The gray matter occupies the center while the white matter occupies the boundary. So in this case you can see that it consists of higher concentration of these cell bodies, basically cell bodies and which are crucial for processing information and forming synapses. So gray matter also consists of this neural body. These neural bodies are also called as neural cell bodies are also called as soma, and it consists of this particular, like all the organelles including the nucleus, mitochondria and each and everything that is essential for a particular cell to function there. And also it consists of these dendrites that carry out these communication aspects and all between different neurons, and also it consists of shorter axons and are basically non-myelinated in nature in case of this case. As case of white matter, it is myelinated. In case of gray matter, majority of them are shorter and are unmyelinated in nature. So basically these receive signals from other neurons, playing a vital role in synapse connections and communication with the nervous system. So the function is quite similar in this case, but the abundance of what is present within that particular matter, what is present with higher in the white matter and what is present higher in gray matter, actually different. But the only distinction is that, or the only similarity is that it is used to carry out this communication or electrochemical sensing and then transmission of this signal to brain and all. So I think this is clear. So anyone has any doubt? Okay, so I think the answer is quite clear. Gray matter is mostly made up of cell bodies, dendrites and shorter axons, while white matter is made up of myelinated neurons. So coming up to question number 3. Color of white matter comes from first option, nucleus, generation of action potential, lipid content of myelin, all of the above. So I think we have discussed the answer, but I expect answers. So what would be the answer to this question?
So lipid content of myelin.
Yeah, so the answer to this question is lipid content of myelin. I think you are trying to speak something, right? I can't hear through my microphone, I think. So I'm not sure if whether from my issue or not. So I think it's better to type it in the chat box, or yeah. So the answer to this question is lipid content of the myelin. So basically, if you look into white matter, you can see that, as I have discussed below, this is the myelinated one, right? You can see the presence of Schwann cells here, and this is the unmyelinated one. You can see that there are no like Schwann cell there or myelin sheath there. So basically, if you look into the white matter, most of them are composed of this type of neurons, that is myelinated ones. And this myelin sheath actually carries a large amount of lipid content there, right? So as I said before, in case of peripheral nervous system, Schwann cells are the ones who is responsible for forming the myelin sheath. But in case of central nervous system, the function is actually carried out by the presence of oligodendrocytes there. So basically, because it's actually those Schwann cells which are individual cells that are combining together and then form a sheath that wraps around the axon to form what is called as myelin sheath there. And these individual myelin sheaths are produced by several groups of Schwann cells there. So because of the presence of high lipid content of myelin, basically they appear most probably white, and that's why that portion of the central nervous system or the brain or the spinal cord is called as a white matter there. And it insulates the axons and facilitate rapid signal transmission between different brain regions and between the brain and spinal cord there. So you can see that the signals are coming out from the dendrite, then it enters the cell body, then it is transported across this region linearly and to the other nerve endings, right, where it transfers from synapse to the next neuronal cell or whatsoever. So you can see that in this particular region, right, there is a particular node there, particular small space there, which is obviously called as a node of Renvir there. So basically the signals that are coming out, instead of passing bilinearly in case of unmyelinated neuron cell, in this case the signal that is received here jumps from this region to this region, then again to this region, then again to this region, and then it transfers to a nerve endings there. So this particular type of conduction is called as solitary conduction, and is much more rapid as compared to this particular unmyelinated one. So so there is basically a difference in this both cases, in this unmyelinated and myelinated neurons there. So yeah. So the answer to this question is the color of white matter comes from lipid content of myelin. So I suppose it's clear. So I think I will just move on to next question. So do you have any doubt? So I think the answer is quite clear, right? It's a basic one, and we have discussed it from the previous question. So I will just move on to the next question then. Coming on to question number 4. What day of embryonic development should motor neurons be dissected for cell culture in rat? Option number 1, E10. Option number 2 is 14, E18, and E24, which might be the answer. I think it's preferable to, if you know the answer, just type in the chat box. Like as previously, I cannot hear you basically. Yeah, so I will just tell the answer then. So the answer to this question is E4, and E stands for embryonic day or embryonic development in this case. So basically, if you look into a lifespan of a fetus or a gestation period means that the day from the implantation of the embryo happens within the uterus of the mother to the day where the organism or the baby is born. So that is called as the gestation period, right? So basically, if you look into a gestation period or embryonic day, you can see that in case of rat, I think it starts from E0, 0th day where the implantation happens, to the E20, or E24th day. So there is a gap, sometimes E20 or E24th day is a preferable day when the baby is born. That is the postnatal. Afterwards the postnatal stage happens, right? So postnatal means after the baby is born there. So in this case, so if you look into the aspect, different stages of development for different organs or different portions of that embryo take place differently according to each and every day, right? So sometimes some portion of the embryo will develop within this particular number of days or embryonic days. Sometimes other portion will happen much later or much before that also. So in case of this hippocampal development or any kind of neural development, nervous system development and all, like E40 is the preferable day where hippocampus is completely developed and hippocampal neurons are completely developed or motor neurons are completely developed there. So basically if you look into the motor neurons are basically present within the spinal cord or peripheral neural system, right? So basically the nervous system is completely developed there. So there are motor neurons that are present there basically. So if you look into E14th day, it is possibly the best day where you can extract the motor neuron from the fetus. So you have to euthanize or sacrifice the mother, then you have to extract the fetus from the uterus of the mother, then you have to do some surgical procedures, then you have to take out the spinal cord or hippocampal cell, whatever is necessary, in this case the spinal cord where motor neurons are present. Then you have to extract them out at this particular day itself, that is the 14th day of embryonic development or embryonic day. So afterwards it is not that possible because sometimes if you see like afterwards what happens is that the neurons start to undergo extensive axonal extension or developmental apoptosis. Basically they are just getting degraded out after E14 because up to that particular stage they are actually dividing, then it starts to get die out or it undergoes apoptosis or programmed cell death, right? So basically at that particular time period you cannot after that particular time period, that is the 14th day, you cannot actually extract out these particular motor neurons because they start to undergo apoptosis. And also it also indicates that at this particular day, that is the 14th day of embryonic development, high yield and purity of these motor neurons can be like obtained at that particular time point itself. So I think this answer is clear. So is there any doubt regarding this, or should I explain it once more? If you have any doubt you can ask, so no issue, you can just type in the chat box on also. So I think it's quite clear basically, so I will just move on to the next question then. Coming on to question number 5. So based on your answer from question number 4, the motor neurons are not preferred to be collected after the day because of, like we have discussed this answer, which might be the answer. From the previous explanation that give, the answer is there basically. First one, the excessive programmed motor neuron death reduce the yield. Embryo dies after the day. Spinal cord of the embryo cannot be accessed after the day, all of the above. Okay, so I think I will just give you the answer. So the answer to the question is excessive programmed motor neuron death reduce the yield. So you can basically see that in the previous slide, right? After the embryonic day 4, where the motor neurons are to be extracted, if you follow up to the next day, then it undergoes extensive axonal extension or developmental apoptosis. It start to die out. So in that case, basically you cannot because of this the programmed death is also called as apoptosis, right? So if it is unnatural death or something of the cell, then it is called as necrosis. So programmed death or apoptosis happens, which actually reduces the yield. So you cannot go beyond this day, that is the 14th day of gestation or the 14th day of embryonic developmental. So after that embryo dies after that day, like it cannot happen because you have to form a mature organism, right? So that embryo cannot die out on that day. So spinal cord of the embryo cannot be accessed after that day. Spinal cord of the embryo can be accessed after that day. There is no limitation to that. So the answer to this question is the motor neurons are not preferred to be collected after that day because of excessive programmed motor neuron death or apoptosis, which reduces the yield. So basically, I think this is clear. So can I move to the next question, or any doubt is there? So I think the answer is clear. I will just move on to the next question then. Coming on to question number 6. Dorsal root ganglion is option number 1, cell body, collection of myelin sheath bundle, Purkinje fibers, or collection of neurons. Which might be the answer? Dorsal root ganglion. Okay, so I will just give you the answer. So the answer to this question is collection of neurons. So basically, if you look into dorsal root ganglion, or DRS, abbreviated as DRS, basically it's a collection of neurons. So if you look into our spinal cord, the spinal cord is actually located on the ventral side of the body, right? The portion of where our ribs actually come out to be, that is called as dorsal side. If it is on the back side of our body or near to the spinal cord, then it is actually called as a dorsal side. So in case of rat also, if you look into the dorsal side where the spinal cord is present, at the E14th day, that is the 14th day of embryonic development, at the lateral side. So if you look, this is the spinal cord there, and if you look at the lateral ends of the spinal cord, several neurons are present there, or a collection of neurons are basically present there, or a collection of cells are basically present there, which represents the dorsal root ganglion there. So basically it is present, as I have said here, each spinal nerve, that is a spinal cord, has a pair of dorsal root ganglia, which if you consider this as spinal cord, one pair is present here, one pair is present here at the lateral end of the spinal cord there. So dorsal root ganglia is situated laterally to the spinal cord, and they play a significant role in processing the sensory input, especially the sensory information like pain, temperature, touch, and everything is maintained because of the presence of this dorsal root ganglion there. So basically it is also a cluster of sensory neuronal cell body. It is sensory in function, located in dorsal root of spinal nerves, which are crucial for transmitting sensory information from the peripheral nervous system, because spinal cord actually forms a part of the peripheral nervous system, right? So this dorsal root ganglion is present at the spinal cord, so it is a part of this PNS there, that is peripheral nervous system there. So after getting the sensory information, it travels to the spine, then it enters into it is transferred to the central nervous system or the brain there. So that's why it is helpful in transmitting these particular sensory information from the PNS to the CNS there. The cell body within the DRG is first order sensory neurons. Their primary function is sensory in nature and are characterized by pseudo unipolar neurons. So basically neurons are of different types. One type is the myelinated or unmyelinated neurons, which I have discussed earlier, and the second is the based on the number of axons that are present there. It can be unipolar, bipolar, multipolar, or pseudo unipolar there. In case of unipolar, if the cell body actually consists of only one axon protruding from it, it is called as unipolar. In case of bipolar, if you look into a cell body, one axon is if this is the cell body, one axon is protruding from there, one axon is protruding from there, or the other axon is protruding from there, then it is bipolar, because there are two different axons that are protruding from a single cell body there. Multipolar, there are multiple axons there, as you can see. And also in case of pseudo unipolar, basically in case of pseudo unipolar, if you consider this as cell body, only one axon is protruding from the cell body, but this axon later develops into two. Basically it is like one single axon there, so you will think that it's unipolar, but it's not, because the axon later on divide into two different axon bodies there. pair of actions there. So it acts as a unipolar, but is actually not a unipolar. So that's why it's called as a pseudo-unipolar neuron there, meaning that they have a single action that bifurcates into two different branches there. So this ganglia serves as a relay point for, as I discussed, sensory information, where the neurons convey signals from various receptors to the spinal cord and the brain. So basically, like, yeah, that's their probable function there. So dorsal root ganglia is basically a collection of neurons. Purkinje fibers, actually, I think it's present in our heart, right? So pacemaker of our heart, I think that's what it means basically. I don't also actually recall there, but I think it's carry out action potential to keep the heart beating there, right? Collection of myelin sheath bundle is not the answer, so answer is collection of neuron. So I think this answer is clear. So anyone has any doubt, you can just type in the chat box if you have any doubt. Okay, so I think this answer is clear. So dorsal root ganglion is a collection of neurons. So I will just move on to the next question then. So coming on to question number 7. Which should be added or what should be added to the medium to prevent contamination? First option is glutamax, antibiotic or antimycotic, B27 or dexamethasone, which might be answered. I'm getting option number B. Yeah, so you are right. Basically the answer to the question is antibiotic or antimycotic. So this is not I, this is Y basically. So if you look into the antibiotic or antimycotic, basically in order to prevent contamination, you need to prevent the growth of any unwanted microorganisms like bacteria, fungi, or more rarely in case of virus also in this case. So basically antibiotic means antibacterial, right? So basically bacteria can be inhibited in two ways, or any type of organism can be inhibited in two ways. Either it can be bactericidal or bacteriostatic. If we are killing the organism as a whole, it is bactericidal using that particular chemical or any substance or any drug you are using, it is bactericidal. If you are arresting the organism, growth of the organism there, then it is bacteriostatic, because you are not completely eliminating the organism there, however you are reducing the growth level of that particular organism there, so that the growth is getting arrested there. It is called as bacteriostatic. Similarly, in case of mycotic, which means that it's something related to fungus, the name actually related to fungus, mycota, that particular name, right? So it can also be like fungicidal or fungistatic, right? So that's in the case. So it is basically Y in this case. So if it is antibiotic, several antibiotics like what you say, one of the common like penicillin is there. Penicillin is used, most probably used in medical application. Ampicillin is used in this case. Chloramphenicol is also used. Yeah. So these are basically antibiotic drugs basically. In case of antimycotic drugs, one of the examples include, there are different class of antimycotic drugs like polyenes are there, sterols are there. If you look into polyenes, amphotericin is one such drug, amphotericin B, which binds to the ergosterol that is present on the plasma membrane of fungal organism, because in case of fungus, one of the common factors is the presence of chitin, which is present in cell wall, and the other one is the presence of ergosterol, which is present in the plasma membrane, right? So this amphotericin B or polyene drugs actually bind to ergosterol. Once it binds to ergosterol, it actually creates pores within the plasma membrane, so the cell contents actually move out of the cell to the external environment and it lyses out the cell. So that's its function basically. While in case of azoles actually go into the endoplasmic reticulum, where ergosterol biosynthesis actually take place. It inhibits one of the or two of the proteins that are involved in ergosterol biosynthesis, like ERG1, ERG11. So these are like proteins that are involved in ergosterol biosynthesis. These move into a endoplasmic reticulum, binds to it, and then inhibits its activity, thereby killing that particular organism there. In this case, the fungus there, right? So basically, these are some kind of drugs that are usually used to prevent contamination, because it kills off this particular microorganism, that is what we have said, bacteria, fungus, virus there. So in case of coming on to the first option, glutamax is the first option. Basically, we have discussed that last week or previous week question, I don't actually recall. So glutamax is actually like a dipeptide of glutamine. Basically, it was said that in the lecture, basically it's a dipeptide of glutamine, but based on what I have read, it's actually a dipeptide of alanine as well as a glutamine. Basically, I am not sure whether first it was actually a preparation of dipeptide of glutamine, later it was turned into a preparation dipeptide which is made up of alanine or L-glutamine. I think you should also check that out. So basically L-glutamine is an amino acid supplement, right? So it is useful for energy source because cells are rapidly dividing, basically protein synthesis and all. Basically you need this L-glutamine and all. And it is also used in the production of purine or pyrimidine nucleotides, glutathione, glutamate, other amino acids, and also involved in, as I said, protein synthesis and also in energy production like glucose production and all. So in case of aqueous solution, this L-glutamine can degrade, generating toxic ammonia, which can kill off the cell culture there. Basically, it will halt the growth of the cell there because it is toxic in nature, ammonia. A high amount of ammonia is toxic in nature, and other carboxylic acid as byproduct. So what it is was done that dipeptide, in this case, I will say that L-alanine and L-glutamine was hydrolyzed to form a dipeptide, which was later named as glutamax there. So basically it undergoes one release of one molecule of water there, so it forms a dipeptide there. So which are more stable in aqueous solution and does not spontaneously degrade there, because in case of sometimes the cell culture has to be stored longer there, right? So the compounds that are present within the cell culture should be stable enough to do that basically. So glutamax is basically in this dipeptide form, which actually quite stable there, and instead once it enters the cell releases aminopeptidides, hydrolyzed cell, and then it releases this L-alanine and L-glutamine into the culture medium, so that it can be actually be taken up whenever it is needed there. So much of them will be present in the dipeptide form, some of them will be released out into L-alanine and L-glutamine, which can be uptaken by a cell to carry out its growth functions and all. So if you look into the B27, which was also we have discussed last time, I am just recalling the what are those and all. So basically it is also called as neuronal neurobasal media, NP27 or B27 it is also referred to as. So basal media 27. Basically 27 represents that it consists of about 27 components within that particular media there. So basically it is used to like in case of growing this neuronal cultures and all. So neurobasal media or NB media is used to culture fetal neuronal cell, while another preparation called as NBA neurobasal A media is used for culturing adult neuronal cells also. This is because in order to grow a adult neuronal cell, it prefers a media containing higher osmolarity as to preferred by the fetal neuronal cells there. So this consists of higher osmolarity there because of the presence of higher concentration of salts and everything. And also basically this particular media as compared to simple common neurobasal media is used for growing adult neurons only. And it also consists of sodium bicarbonate, which buffers the pH there, and also other compounds like amino acids are provided, vitamins, inorganic salt, glucose, phenol red, etc. So these are the basic function of a neurobasal media. Dexamethasone, which is the last option there, you can see from that. It is actually a glucocorticoid. Basically, it's a corticoid drug that binds to certain receptors that in case of our body, it is used to reduce the inflammation once taken into our body. So in case of cell culture, it somehow affects the proliferation, differentiation and apoptosis. So it is not involved in this by preventing contamination and all. However, it supports these kind of activities there. So yeah, so I think this answer is clear. So does anyone have any doubt? Okay, so I think it's clear. So I will just move on to the next question then. Coming on to question number 8. Density gradient centrifugation is a method of cell separation, cell doubling, cell addition or organelle culture, which may be the answer. Density gradient centrifugation or ultracentrifugation. I am getting option number A. Any other option? Yeah, so the answer is right, you are right basically. So option number 1, that is cell separation. Basically, density gradient ultracentrifugation operates on the principle of sedimentation under centrifugal force, where the sample particles in the sample are separated based on their density. You can see that this is the density, the gradient there. You can see that in case it is lighter, in case it is darker. So the darker area means that if you are using a component like a sucrose, or I will explain this. Basically, I will just give you the principle. So in case of all type of centrifuge, basically a centrifugal force is there, right, which act outwards. So outwards from the point of the origin. So basically, when you are spinning a sample or in a centrifuge at a higher centrifugal force or a higher RPM, you can see that the particles start to settle, start to move onto the end of the falcon or the test tube there, right? So this is basically because of the presence of a centrifugal force there that makes the particles move from the point of origin to the end region there because of the centrifugal force that is acting outward to the radius of gyration there. So because of this, the denser particles start to move away from the much distance away from the origin, while the less dense particle does not move that much, but will be much or more closer to that of the point of origin. It doesn't move that much as compared to the highly denser particles there, because of the size of the particle, shape of the particle, whatever this density of the particle most probably in this case. So basically in this case of, so that's the principle there. Based on the presence of centrifugal force, particles based on the density, size and shape will move from the original point to a much outer point away from the origin where it is where the origin of rotation there. So basically in this case you are providing a density gradient median there. Like components like sucrose, cesium chloride are used and are prepared. Basically if you look into this image there, you can see that it's much darker, meaning that if you are using a sucrose as the density gradient provided there, you can see that it is much darker as compared to much lighter there. Here the sucrose gradient is much higher, as concentration is much higher as compared to the top region here. That's why the density of the sucrose is actually getting varied there. So here the concentration of sucrose is higher, then it starts to decrease over time. It starts to decrease up to this point, and the sample is loaded at this top at this particular point there. So basically if you are increasing the concentration of the sucrose there, then the portion of this particular gradient or this particular layer much higher density as compared to the top where it has much lower density there or much less density there. So particles which are less dense actually occupies this less dense area, while particles which are much more denser when they are kept in a centrifuge moves downwards with increasing RPM there, and based on their density. So if a sucrose has a density gradient over here, like if I am saying that it has about 1 unit of density there, and if the particle that is moving across this has also the 1 unit of density there, then we will occupy this region there, because the density of the particle is equal to the density of the sucrose gradient there. If it is much more higher, in this case you can see that the particle will have a much higher sucrose density in this case, and the particle will occupy this particular region itself. So you can see that smaller size particles actually occupies at the lesser concentration of sucrose gradient there, while the medium sized particle comes in between, while the large size particle actually occupies at the bottom because it is much more denser as compared to the other particles there. So that's basically principle there. So isopycnic point is also a point you have to remember, that when the gradient in reaching the density will match that of the surrounding medium, then it is called as an isopycnic point there. At this point, the net forces acting on the particle are equal. So that's why the particle will stop moving, and then it will occupy its corresponding density there, because the density of the sucrose or the gradient there is equal to the density of the particle there. It will stop moving, then occupy that particular space there. So layer formation will be also there. Denser particle will accumulate at lower level, while the less dense particle will occupy much above them. This helps to like separate out heterogeneous mixture there. So in this case, it is used to separate out cell separation. It is used to carry out cell separation there. So I hope this answer is clear. So any doubt is there, or should I move on to the next question? Okay, so I think this answer is quite clear, so I will just move on to the next question. Coming to question number 9, crude trypsin is more effective, show more effective activity than pure trypsin because presence of other proteases, presence of other amino acids, presence of lipids, or presence of fibroblasts. Which might be the answer? Okay, option number A. Yeah, so you are basically right. The presence of other proteases basically there. So it's not the pure trypsin, you have to know, it's crude trypsin. So in case of crude trypsin, majority of the concentration will be of trypsin itself, but it doesn't mean that other components will not be present there. So that's the difference between crude as well as a pure trypsin there. Pure trypsin is purely made up of trypsin, while crude trypsin is concentrated amount of trypsin there, but other substances are also present there. So in this case, if proteases are present there, it will definitely increase the activity because if you are using any crude trypsin to remove extracellular material for cell separation and all, it is not preferred because the presence of other proteases will increase the activity of extracellular matrix. So it will first degrade out the extracellular matrix, then it starts to degrade out cells there. So basically we don't want that because of the higher amount of activity there. But in case of pure trypsin is not there. If you are putting it up to like 15 to 20 minutes or 10 to 15 minutes, it will only clear out the extracellular matrix, separating out the cell. Then you can inactivate the trypsin there using several inhibitors called as trypsin inhibitors there. So basically that's the activity there. In some cases, presence of other amino acids are also important because some amino acids are capability to activate certain proteins even they are present in pure forms and all. So it's quite spectacle, but it's not that much of amount, but due to presence of the other proteases, the activity of the crude trypsin is more active, show more effective activity than pure trypsin because of the other presence of other proteases there. So any doubt here? Okay, so I think this answer is quite clear. So I will just move on to the next question then. Yeah. Crude trypsin exhibits more effective activity than primary trypsin. That is all that I explained there. So yeah, so I will just move on to the next question. Question number 10. Skeletal muscles produce GFAP, myosin heavy chain, MAP2 or neuron. Which might be the answer? Skeletal muscles. Okay, option number B. Yeah, so the answer to this question is myosin heavy chain. So basically myosin also consists of heavy as well as light chain there, right? So we are only explaining this in this case. So basically MAP2, you know what MAP2 is? Basically we have discussed that in last class if you might have joined. It's something associated with neurodegenerative disorders and all. So basically it is also called as microtubule associated protein 2. Basically it actually regulates the cytoskeleton that is found in like hippocampal neurons and all. So and also it helps in microtubule or assembly and everything. So basically these are proteins that are found that are endemic to this particular pyramidal neurons. In some cases it is sometimes found in other neuronal culture there, but in case of hippocampal culture that is this pyramidal neurons and all, it is actually associated with that. So basically for your information I'm just recalling that. So basically myosin if you look, it has two chains, heavy as well as light chains there. So this heavy chain has a globular head there, right, which helps in the binding to actin there. So I think I will just move on to the next slide there. So you can see that this is a myofibril. This is the single elemental unit that represents a muscle fiber, right? So if you look into that, there is myosin there, and this is the actin filament there, and this is another protein called titin, which helps in the in case of muscle when are in relaxed stage, this titin helps in the attachment of this myosin to this particular end of the region of the myofibril there, because in order to maintain the structural integrity, all the proteins have to be present within that range there, just like how an extracellular matrix actually keeps cells together there. So basically if you look into this is the myosin, this is the actin filament there. So this is the actin, this is the myosin. So it consists of a light chain as well as a heavy chain, and the heavy chain consists of a this particular protrusions there, globular head, we usually call it as myosin heavy chain globular head, right? So this head region actually binds to the I think the tropomyosin region of this of this actin filament there, right? So upon binding, ATP hydrolysis as well as dehydrolysis happens, and because of the subsequent hydrolysis as well as dehydrolysis, the muscle has a tendency to relax as well as contract. When it is contracting, the myosin actually binds to this actin filament there, then it moves inwards there. So if you look into this region there, then it moves inwards, so it contracts into the M line. So this particular region, this particular region, distance between these two ends actually decreases out. So in case of relaxation, because of the ATP hydrolysis and dehydrolysis there, you can see that the myosin actually moves away. So this particular distance actually increases there, and because of this particular sequential motion, the muscles are able to contract and relax. So that's why myosin and actin are important components of what constitute a myofibril there. Myofibril, as I said, is a single most elementary unit of a skeletal muscles there. So I think, and this particular portion, if you consider as a whole, it is also called as a sarcomere, and each and every term represents a muscle fiber there. So do you have any doubt regarding this? It's a basic one. You might have seen that in your UG, PG and everything like that. So I think this is quite clear. So I will just move on to the next question there. Coming on to the next question question number 11. Astroglia is used to maintain cellular homeostasis, or immune cells of the brain form myelin sheath, or none of the above, which might be the answer. Yeah, you are right basically. So option number A, maintain the cellular homeostasis, or cellular homeostasis regulation and everything. So astroglia is also called as astrocytes, and therefore part of central nervous system, not of peripheral nervous system but of central nervous system. So I have already told that this is the oligodendrocyte, right? In case of peripheral nervous system, Schwann cells helps in the formation of myelin sheath there. In case of central nervous system, oligodendrocytes are the ones who are enabling the formation of myelin sheath there. So as compared to Schwann cells, which combine together to form a sheath-like structure that wraps around the axon, these oligodendrocytes actually form protrusions from its cell body. Then it attaches itself to the axon at particular points, leaving behind this particular node region called as nodes over and where there, forming this particular myelin sheath in case of myelinated nerve fibers and all. So this is the function of the oligodendrocyte, which carries out the formation of myelin sheaths there. So in case of astrocytes, you can see that this green colored cells, right? So basically it helps in the supporting the formation, it does not form myelin sheath, but it also helps in the supporting the formation of myelin sheath, which is carried out by oligodendrocytes there. Apart from that, this is actually involved in regulating the cellular homeostasis. How? Because they help to control the iron balance, remove excess neurotransmitters like glutamate, and also provide metabolic support to the neurons when they are getting developed there. So this is how it maintains the cellular homeostasis there. Somehow some amount of function is also involved in the formation of myelin sheath, but it's totally the activity of oligodendrocyte there. And in case of astroglia or astrocyte, it is involved in maintaining the cellular homeostasis there. So yeah, so I think the answer is clear. So anyone, do you have any doubt regarding this? So I think this is clear basically. It's one of the example of glial cells that are present within the central nervous system. Okay, so I think it's quite clear. So I will just move on to the next question. So in this case, astroglia maintains cellular homeostasis. So coming on to the next question, question number 12. Microglia maintains, option number 1, maintains cellular homeostasis. Are immune cells of the brain form myelin sheath, or none of the above, which might be answer. Yeah, so the answer to this question is option number B, are immune cells of the brain. So basically, if you look into a previous picture, this is the same picture that you have shown here. You can see that there is a oligodendrocyte there, there is a neuron there, there is the astrocyte there, which helps in cellular homeostasis, and oligodendrocyte which helps in the formation of myelin sheath there. Microglia acts as an immune cell of the brain there. So basically, microglia acts as the brain's primary immune defense. They monitor the central nervous system or CNS for pathogens and debris responding to inflammation, and can exhibit both protective and damaging role depending on the contents there. And they are also called, because of this particular function, because like similar to the function of what a macrophage does, or what a B or T lymphocyte does, right, or a majority of the lymphocyte does, microglia has the same function there. So that's why it is called as called as a brain's macrophage, because even though this has several protrusions that are coming out of his body, they are actually phagocytic in nature. Phagocytic meaning like it has a it can form an irregular protrusions out of the body, similar to what you see in a macrophage or an amoeba there, because it can undergo the phagocytosis, right? Amoeba undergo phagocytosis to engulf in food and all. In this case, microglia can undergo phagocytosis, or irregular protrusions are forming out of the body to engulf in this particular pathogen, debris, etc., and to carry out this particular immune defense function that is particularly absent in case of brain and all. So yeah, so I think the answer is clear. So that's why it is also called as I repeated earlier, brain's macrophage. So microglia are also called as immune cells of the brain. So any doubt in this case? Yeah, so I think it's quite clear. So microglia are the immune cells of brain, and also called as brain's macrophages, because it carries out this whole immune defense functions and all, and has a capability to undergo phagocytosis. So I will just move on to the next question. Question number 13. Density gradient centrifugation utilizes a column with dash density top from bottom. First option is decreasing, increasing, same, none of the above, which might be the answer. Density gradient centrifugation, we have discussed this before. Yeah, so the answer to this question is increasing basically. Yeah, you are right, basically increasing, option number 2. So basically I am just repeating the answer here. So basically this is the Falcon tube, or the ultracentrifugation tube. If you are carrying out a tube, there are specific tubes called as ultracentrifugation tube. Otherwise, under the high centrifugal force that are created in the ultracentrifugation, as compared to a normal centrifugation will be high, the tubes have a tendency to break down, and the sample will be spilled all over the centrifuge there. So these are specific tubes there. So in this case, you are filling in different layers or gradients of whatever gradient material you are using. Suppose that I am taking here, in this case, what is it, sucrose there, or sucrose there. I am taking in sucrose there. So I am having a higher concentration of sucrose there. So it is much more denser in this case. Then I am having much more lesser concentration, and the concentration actually decreases from when it moves from top to from bottom to top. In case of top to bottom, this is the place where having the lowest density there, where this is the place having the highest density there. So density actually increases from top to bottom. So that's why the answer came out to be increasing from density from top to bottom. Because, and this density change is basically due to a change in the concentration gradient of sucrose there. So as I said, in this case, the density is less. As the density of the layer is less, it has less sucrose content or sucrose concentration as compared to this region, which has the higher sucrose concentration, which maintains the higher density gradient there, and this uniformly changes throughout. So the density from top to bottom starts increasing because of the higher levels of sucrose that you are providing there, and based on the density gradient there, if you are loading any sample into that, heterogeneous sample into that, based on the density of each particle that is present in the sample, they have a tendency to localize into this particular regions there. So smaller size particle having lesser density occupies a much lesser gradient there, or concentration gradient there, or density gradient there, while medium size particles come in between the large size particles, and the large size particles actually occupy the lowermost region where the density of the sucrose, in this case which I am using, is much higher. So the density gradient centrifugation uses a column with the increasing density from top to bottom. So do you have any doubt regarding this? We have discussed this in the previous question also. Yeah, I think the answer is quite clear, so I will just move on to the next question then. So coming on to question number 14. Which of these is not a cell of nervous system? First one is neuron, second one is astroglia, third one is Schwann cell, and fourth one is nephron. Which might be the answer? Option number D. Yeah, you are right. Basically, nephron is a part of kidney, right? It's the single most elemental unit that is important in kidney, nephron, for like blood filtration and everything. So basically, in case of neuron, you know the answer. Astroglia or astrocytes are basically the cells of the central nervous system that actually regulates the cellular homeostasis by maintaining the ion conductance, presence of neurotransmitters along the synapse and all, right? So basically, it regulates the cellular homeostasis there. So Schwann cells, basically these are present within the peripheral nervous system, not in the central nervous system. So basically, it has a critical role in regulating, supporting, and insulating the neuronal axons there. So this is the neuronal axons there. There is Schwann cell, and these are the myelin sheath there, and these are the regions between two Schwann cells. Part of the axon between two Schwann cells are also called as node of Renwier there. So basically, it is coined out by the scientist Theodor Schwann, and helps in the formation of myelin sheath within the peripheral neurons there, and which enhances the speed of the electrical conductance that are taking place between these nerve fibers there. So there are basically two types of Schwann cells, two types of neurons there. In case of Schwann cells, if it is myelinating, it's called as myelinating. If it is non-myelinating, it's called as non-myelinating there. So in case of myelinating Schwann cells, or in case of myelinated neurons, each Schwann cells wraps around the single axon to form a myelin sheath there. And basically there are several Schwann cells in the presence of different proteins like myelin basic protein. Some of the boundary of these Schwann cells are lost there. When this boundary is lost there, they aggregate together to form a sheet-like structure. Then the sheet-like structure actually wraps around the axon to form a myelin sheath there. Then another sheet-like structure wraps another region, forming a myelin sheath near to this region itself. So this is how a myelin sheath is formed by the Schwann cells, and this is unique to peripheral nervous system itself. So this structure significantly reduces the mechanical membrane conductance and increases the conduction velocity, and this particular process is called a saltatory conduction there. So if you look into a conduction aspect, you can see that the signal coming into a cell body, then it is transferred to this region, right? In case of non-myelinated, it will pass along the axon, then it moves to other nerve endings there and passes on to another neuron itself. In this case, if myelin sheath is present there, then the signal that is reaching here is jumping over one myelin sheath to the nodes of Renwier. Another myelin sheath is coming there, and then it jumps once again, then it jumps, and this process continues until it reaches the nerve endings there. So this is the basically process called as the saltatory conduction there. So this is much more higher velocity as compared to a normal conduction that is taking place in non-myelinated neuron cells there. So in case of non-myelinated Schwann cell, they provide support and nourishment to myelinated axons, but it doesn't involve the formation of Schwann cells there. So I think this particular explanation is clear. So do you have any doubt? If you have any doubt, you can just type in the chat box, no issue, because I think there is an issue with the audio here. So yeah. Okay, so I think this question is quite clear. So I will just move into the next question. So which of these is not part of central nervous system? Nephron is the part, is part of a kidney or filtration system. So coming on to question number 15. Which of these is a recommended density gradient medium? Option number 1 semi-prep, option number 2 semi-osmo, opti-prep or opti-osmo. Which may be the answer? Yeah, option number C, that is option number 3, opti-prep. So basically if you look into like different density gradient medium, right? So in case of density gradient ultracentrifugation, we know that the density gradient should be there, so the particles or cells can be separated out based on their density, right? So basically the commonly used ones or traditionally used ones are sucrose and ficol or cesium chloride there. So nowadays opti-prep is also a component that is being used there to make the density gradient medium there. So basically it is sterile in nature. Whatever we are using should be sterile, shouldn't consist of any other particle or any endotoxin, especially in case of cell culture and all. You are adding that into a particular falcon or a vial that consists of this density gradient solution there, right? So it should not consist of any endotoxin. If there is any endotoxin or any other substance that is toxic to cell, then the cell will degrade out over time and your all work will go into waste. It will take some time there basically. So in this case it consists of a solution of iodisanoate instead of sucrose or cesium chloride or a ficol. Basically it is provided with this particular compound iodisanoate. I'm not also much aware about this particular compound, and it is used in separating out, especially in isolation of cells and other organelles there. So basically it is sterile and endotoxin tested there. And opti-prep, basically if you look into opti-prep, this particular compound iodisanoate has a tendency to maintain the isoosmolarity there. Isoosmolarity means that there is not much osmolarity fluctuation there. So if you are providing a cell there, then the osmolarity of that particular cell will much more or less be similar to that of the iodisanoate gradient, density gradient there. So basically the density changes will not affect the osmolarity capacity of that particular gradient there, and that's why that's why one of the most important aspect of using this particular opti-prep is because of this particular capability of maintaining the osmolarity there in this case without affecting the cells as a whole. And also it also has the it also has lower toxicity as compared to other compounds that are used to make a density gradient medium there. So that's another reason, and also it also has the capability to make a uniform gradient medium on its own. So in case of sucrose, you have to make different concentration of sucrose there, then you have to put it into layer by layer. So in case of this compound, it has a capability to form a density gradient there. Automatic density gradient is available for this particular media itself. So one component is the presence that it has the capability to maintain the osmolarity there. Second is it has a capability to make a formation of a continuous or a discontinuous gradient there. And also it has much more lower toxicity as compared to other gradient production medium that has been used there. So I hope this particular answer is clear. So do you have any doubt? So I think the answer is quite clear. So the answer is which of these is a recommended density gradient medium? Third option, that is opti-prep, which is made up of this particular compound iodisanoate. So I will just move on to the next question. Coming on to the next question, this is a theoretical question. Suppose based on your answer in question number 5, what is the density of a required density gradient medium? First option 0.32 milligram per ml, 0.32 gram per ml, 1.32 milligram per ml, or 1.32 gram per ml, which may be the answer. Which is the recommended density gradient, density of a recommended density gradient medium. Okay, so I am getting option number D. Yeah, so the answer to this in is option number D, that is 1.32 gram per milliliter. So basically, even if you are like carrying out a density gradient centrifugation, right? So if the density of the medium is not like within a recommended value, chances are that the cells that you are adding into the mixture, the changes in osmolarity will subsequently happen, and that will like affect the cells also. So there is a recommended density that is required for generation of each density gradient medium that should be followed, and because of this presence of like particular and this, when you are making a density gradient medium, the density gradient or the density that should be prepared from that particular density should be followed there. So suppose that you are preparing a density of about 1.32 gram per milliliter, then you are lowering the density upon subsequent dilution, right? Then this subsequent dilution, the one with the highest density is added into the centrifugation tube at the first, then the lower density, the subsequent serial dilution that have been made is getting added, forming a layer by layer, and this layer by layer addition actually forms a density gradient there, right? So the density should be kept in mind. It is approximately about, as it is given here, 1.32 gram per milliliter, and from that prerequisite other subsequent density should be maintained there, because as I said, the cell viability should also be maintained when you are doing cell separation using ultracentrifugation and all. So that's a prerequisite. So I think this answer is clear. So do you have any doubt? So I think the answer is clear. Based on your answer in question number 5, what is the density of a recommended density gradient medium here? It is option number 4, that is 1.32 gram per ml. So coming on to question number 70. So question number 70, that is density centrifugation is based on only buoyant density, only sedimentation rate, both A and B, none of A and B. Which might be the answer? Yeah, option number 3, that is option number C, both A and B. So basically, if you look into this aspect, you can say that buoyant density, first of all, the first option, buoyant density is the contents of density gradient centrifugation refers to a specific density at which particle or molecule will flow with a gradient solution. This technique, also called as isotechnique or equilibrium density gradient centrifugation, separate molecules based on their density differences. So basically, I will move on to the previous slides. Yeah, so this slide. So basically, yeah. So you can see that these particles actually move on to based on the density, right? So basically, if you look into a small size particle, as it is smaller, density will be less, and also in this case, as compared to the latter concentration of the density there, you can see that the particle density of the particle will be or the buoyant density of the particle will be equal to the density of the that particular sucrose gradient that will be present there. So if it has a density of about 1 gram per ml, then the particle will also have a density of about 1 gram per ml. So in this case, if the particle has a density of 1.2 gram, if the sucrose layer has a density of about 1.2 gram per ml, then the particle here will also have a density of 1.2 gram per ml. So here large size particle, if it has 1.3 or so, then it will have a density of 1.3 gram per ml. So it happens like that. The buoyant density of the particle should always be similar to the density of the sucrose layer that is present there, or the sucrose gradient that is present in that subsequent layer there. So that's why the particle stops at that particular gradient and does not move as compared to the other particles having the higher density there. So that's what buoyant density means actually. So it is also called as isotechnique or equilibrium density gradient, because it is something that is related to density there, right? So density differences should be similar in both cases, so the particles won't move according to that. So in density gradient sedimentation rate is also important, because the rate of sedimentation actually dictates the size, shape, as well as the density of the particle there. So based on the varying density, the sedimentation density of each particle will be much different as compared to the other particles there. So this technique is crucial for separating components based on their density, size and shape. So in this question, the density gradient centrifugation is based on not only buoyant density or sedimentation rate, but both in this case, that is both buoyant density as well as both also in case of sedimentation rate also. So I think this answer is clear. So do you have any doubt? So I think this answer clear. I will just move on to the next question then. Coming on to question number 80. So which motor neurons regulate the sizes of muscle spindle or intrafusal fiber? First one is alpha, second one is beta, third one is gamma and delta. Which might be the answer? For intrafusal muscle that is present within a muscle spindle. Yeah, so I am getting option number 3. You are right. So the answer to this question is gamma motor neuron regulates the sizes of a muscle spindle or an intrafusal fiber. So muscle spindle are basically stretch receptors that are present within the skeletal muscle. So this is a skeletal muscle. So basically, if you look into that, there are several different stretch receptors that are present there. So these stretch receptors are also called as muscle spindle. So if you look into a muscle spindle, you can see that there are several different organization of muscle fibers there. So it is basically composed of these two fibers, that is this one the extrafusal, and these particular muscles also called as the intrafusal muscle fibers there. So the motor neurons that regulates the size of this particular muscle, these three muscles that are represented in three different colors. I hope you can see that basically. So these are also called as intrafusal muscle, as intrafusal fibers that are present in muscle spindle that I discussed earlier. So these neurons, these motor neurons that regulates the size of these intrafusal muscles are also called as gamma neurons. So basically, these neurons intervenate between these muscle fibers. You can see that these neurons actually connect to these intrafusal muscle fibers, and based on changes in the stress receptors or the signals that are coming to stress receptors there, these neurons actually helps the muscles to innervate or change its structure there, allowing them to adjust to the sensitivity of the spindle to stretch. So in order for this actually is present within the muscle spindle, right? So in order to muscle spindle to stretch, then this particular muscle fiber should also stretch. So these particular gamma neurons that are connected or interacting with the intrafusal muscle fibers there actually sends out a signal saying that you need to fuse, because the muscle spindle needs to contract there, so you need to stretch out itself. So these particular motor neurons are the motor neurons that are involved in this particular signal type, and also so this mechanism is crucial for maintaining the proprioception and ensuring that the muscle spindle can accurately dictate changes in muscle length and tension during the movement. So that's the function of this particular motor neuron there, because in the name itself you know that it's something that is related to movement there, right? Motor neuron. So gamma neurons attached to this intrafusal muscle fibers that are present within this muscle spindle, regulating itself to stretch there. So I think this answer is clear. So do you have any doubt? Okay, I think the answer is quite clear. So I will just move on to the next question. So coming to question number 90. Which motor neuron regulates the size of extrafusal fibers there? Alpha, beta, gamma or delta? Earlier one we have discussed about intrafusal one, right? This is about extrafusal one. Which motor neuron does that? which may be the answer. Okay, so I am getting the option number alpha neurons. Yeah. So you are basically right. It's the alpha motor neurons that regulate the size of extrafusal fibers there. Basically, these motor neurons are of basically two types, alpha as well as gamma. So one connects to alpha, connects to the extrafusal fibers, and it is much larger as compared to a gamma motor neuron. While the gamma motor neuron is much smaller, it actually connects itself to the intrafusal fibers or the muscle spindle. So basically this, if you look into a structure, you can see that this is a structure that I earlier discussed. You can see, you know the structure, right? So extrafusal fibers are present on the outer edges of this particular muscle spindle, while the intrafusal muscles are present within this within the between the two extrafusal spindles there, right? So I am just zooming out this image there. You can see that the gamma motor neuron itself attaching to the intrafusal muscle fibers there, while the alpha motor neuron attaching to the extrafusal muscle fibers there. So and also to the other end, you can see that also in this case, and this is an enlarged portion of the muscle spindle there. So basically these two motor neurons, that is alpha motor neuron and gamma motor neuron, and alpha motor neuron being much more larger as compared to that of a gamma motor neuron, actually coordinate together, and then based on the stretch receptors that are present in the muscle fibers, it actually helps the muscle fibers to contract by contracting or innervating or extravating, whatever it is, by affecting the activity of those, what is it, that extrafusal as well as intrafusal muscle fibers there, which in turn coordinates to the muscle spindle movement there, causing it to stretch. So and these motor neurons are involved in this particular function also, that is alpha and gamma motor neurons. That is alpha motor neuron coordinates itself with the extrafusal muscle fiber, while the gamma motor neuron coordinate with the intrafusal muscle fiber that are present within the muscle spindle of each skeletal muscle there, to coordinate the function in order for the muscle fiber to stretch. So I think this answer is quite clear. So do you have any doubt? If you have any doubt, you can just type in the chat box, no issue. Okay, so I think the answer is quite clear. In case of the alpha motor neurons are regulate the size of extrafusal fibers, while gamma motor neuron regulate the size of intrafusal fibers, which together coordinate the activity of both extrafusal as well as intrafusal muscle fiber, allowing the muscle spindle to stretch, which in turn affect the activity of the skeletal muscle as a whole. So I will just move on to the next question, last one. So question number 20. What clustering occurs on the myotubes at the neuromuscular junction? First one nuclei, second one acetylcholine receptor, third one cytoplasm, and fourth one astroglia. Which might be the answer? Yeah. So the answer to this question is acetylcholine receptor. I will just open the present then pointer there. Okay. So acetylcholine receptors are the answer basically. So basically these are receptors that recognizes the presence of acetylcholine, which is obviously a neurotransmitter there, right? So clustering of acetylcholine receptors occurs at the myotubes of the neuromuscular junction, and this is crucial for the formation of function of neuromuscular junction, where acetylcholine receptors aggregate at the postsynaptic membrane in response to signals from motor neurons. So basically it is similar to neurotransmitters transmission there. Basically acetylcholine, as I said, is a neurotransmitter, right? So it needs to be transmitted from one neuron to the other end, and this transmission actually takes place between the synapse, right? So basically synapse is a region that joins the indirectly joins the nerve ending of the first neuron to the dendrites or the nerve the dendrites that are present within the subsequent or the second neuron, right? So from the first neuron, the nerve endings transmitter. Then between the synapse, the acetylcholine movement happens, which is a neurotransmitter. That's why it's called as a neurotransmitter there. And then it is received by the second neuron, where signal transmission actually happens there, right? So there is a flow between the transmission that is happening between nerve fibers there. So this particular acetylcholine receptor clustering occurs on the myotubes at the neuromuscular junction there, basically for nervous transmission there. So I think this answer is quite clear. And also, if you look into the other option, astroglia, or as I have said before, astrocyte is a part of the central nervous system, where it helps to regulate the cellular homeostasis, right? Or by maintaining the ionic balance there, or by regulating the presence of other neurotransmitters like glutamate or acetylcholine there. So this particular function is carried out by astrocyte, and also it also provides a supporting function to the oligodendrocyte. Basically, oligodendrocyte, which is present in the central nervous system, actually helps with the formation of myelin sheath there, right? So this myelin sheath is in the central nervous system is formed because of the presence of oligodendrocyte, while this astrocyte or astroglia has a particular supporting function relating to the formation of myelin sheath there. While in case of peripheral nervous system, Schwann cells actually carry out the formation of this particular myelin sheath there, right? So basically, if you summarize this, Schwann cell actually aggregate together to form a sheath which wraps around the axon to form that Schwann cell myelin sheath within the peripheral nervous system there. So that's the basic answer there. So do you have any doubt regarding this? Okay, so I think this is the last question there. So thank you for attending the class. So yeah, I will just switch it off. So do you have any doubt in oral? Like oral, I think is clear. So if you have any doubt, you can just type in the chat box. Okay, so I think it's quite clear. So if you have any doubt, you can ask in the next class also. So thank you for joining and have a nice day.